Okay, welcome everybody to the second session of today, the last day of this meeting. And after this session, we'll have a closing session. So please stay there. And I will now invite Professor Ranjini Bandyopadhyay to chair the session. This session. Please, Ranjini.
Thanks, VK. Thank you. Thank you. So let's get started. The first three talks are 15 minutes. So after the talk start, I will switch off my video, but then I'll switch it back on at about 10 minutes. So you know the speakers know that you have about 2 minutes to wind up, and then we'll keep the last 3 minutes for questions. So can I request the first speaker to share his screen? Our first speaker is Orgho Dash. Thanks, Orgho. And he will be talking about local correlation and extreme values in CD models. You can start now.
Thank you. Am I audible?
Yes, you're audible. You're audible? Yes.
Okay, thanks. So, good evening all, and I start with thanking the organizer. So, today I will be talking about local correlation and extreme values in CD models. This is a work being done with Muskan Slit at Hyderabad. So, the program for the goal is to make characterization of the static and dynamic properties of the so-called fluctuation dominated phase ordering. So this FDPO, this occurs when a system exhibits a macroscopic order as well as equivalent fluctuation. This usually appears in a system where order is evolving in a fluctuating environment. I will give a detailed example. So the main purpose is to understand the Coulson kinetics of FDPO. Here we will use two observables. One is a local cross correlation, which is recently introduced, and another is external statistics. We will see as we go on. So, what is FDPO? How does it look like? So, it is the simultaneous occurrence of macroscopic order and equally large fluctuation. So usually, when we know that when you see there is some order set up in a system, say some particular clustering happening, or saying zero-like models when a macroscopic fraction of the mass condenses in one side, the clusters are more or less stable. So we see there is a chunk of mass and there is some particle going in and out, so the fluctuation is small. However, in FDPO, the thing is not like that. Here, as you can see, this is a this is a picture at different times in the steady state. You can see at some time there are two very large clusters. Next there is one cluster. Then there are actually three clusters. Again one. So the clusters are highly volatile, but still retains its macroscopic shape, but also giving rise to a very large fluctuation. Now the question is, when this kind of order is setting up with time from a random configuration, initial configuration, how does it go through in the coarsening phase? How does it build up? So usually the building up of order that is studied to the equal time to one correlation. So it is understood that as you know, when some random configuration which is getting ordered, so ordered domains are formed, and it is understood that within the domain the things are more or less correlated and the domain is growing. So there is a scaling hypothesis which says that only one length scale is sufficient to tell you about the domain size, and it is characterized through this scaling of two-point correlation, where if you scale the distance by a suitable length scale, time given a length scale velocity, then you find there is a scaling product. And it is known that for usual kind of ordering, this curve, this scaling curve, that behaves in a linear manner, which is Burroughs law. But in FDPO, this Burroughs law is violated. So you see, instead of y, there is what is called alpha exponent. which is less than 1, which signifies the violation of Froude's law. Now, what is violation? So this is usually ascribed to a very special kind of behavior which appears in FDPO. So for example, compare these two pictures. One is the usual phase ordering, where you start from a random configuration which goes through a portioning process, and finally one or two very large chunks are there in the system. And you see these black patches which are particles, and these are say holes or a different kind of particle. So there is a very sharp interface. However, here from a random dimensional configuration, you go through a portioning process, but you can see there are large chunks, but there are also small potholes, which is like a disorder, and the disorder is all over the place. So this disordered interface, which is actually broad, which I will tell later, this is responsible for this kind of behavior where you see the problem is violated. Now, it is recently in last 10-12 years, many shown to exhibit this kind of behavior. So it is interesting to see that how this kind of order can be characterized. So recently it is proposed that since there are, as you have seen, there are many all possible domain sizes, small, big, medium. So it is proposed that to characterize this FDPO, we need infinite number of order parameters. But here we ask this question that when you are the FDPO, that is actually co-occurrence of order and say some sense of disorder, domain words. So whether we can characterize order as well as the disorder, so that things are manageable. So we will try to understand how domains form and how the interfaces also build up. And this will be studied in the context of the so-called coarse-grained depth, the CD models. CD models are basically what I feel is kind of a benchmark of the FDPO, because okay, this is a model where you define an interface which may be an Edward Wilkinson or KKT interface in 1D, which is fluctuating and growing, and you sprinkle some particles on it, and you assume that the particles relax very fast. So whenever the surface tilts, the particles immediately relax to its local minimum. So accordingly, it is seen that based on the definition of the CD model, it exhibits only FDPO behavior. So the definition is you have a surface and you define a reference height, and then you say that everything that is below the reference height is filled up with a particle, and everything above the reference height is empty. So that gives you the domain structure of CD. As you can see in this slide, so this is a fluctuating interface, and these lines are different reference heights. Based on your definition of reference height, you have different CD models. For example, you can choose one particular side of the surface and say that the height of that side is the reference height. So as the side fluctuates, the reference height also fluctuates. Or you can also tell that the mean height is the reference height. That will be the CD3 model. Also, there is a CD4 model. So the details are okay. Now, once you define the reference height and define the domain, then you get this kind of structures. You can see this domain interface structure. So this is how this structure actually came up. Now the question is how to characterize the behavior. So we are saying we will characterize the order, these domains, and the disorder, the potholes. The order is readily visible. It is the domain length. We have a domain size. If you try to understand it, you have some idea about the order. But how to characterize the disorder? So recently, a quantity has been defined, which is a local cross correlation of occupancy and the local curvature. So, for example, if you imagine the surface, it has a localities like this or maybe this, and there is a particle or hole in between. So you define a local curvature and the occupancy, and you define this local cross correlation. Now you take a sum over all the sides, and that will give you exactly the number of intersections. So as you can see here, these intersections are all these potholes. Wherever the cut, actually the line cuts the fluctuating surface, at those points the disorder come up. So basically, you quantify the cuts through this local cross correlation. And in fact, this is for CD model, and in fact, it is recently proposed that this quantity S can characterize a large number of heads. So to study this behavior, this is the steady state behavior of this local cross correlation S, and in the steady state, it grows as 1 by square root L, and this is the scaling behavior of the distribution of S. So as you can see, the full distribution scales as root L. So this is a very broad distribution, and this is a scaling function which is often numerical. We don't have a series structure. Now the interesting question is, how does this behavior emerge? How does the interfaces evolve in time? So as we started with, there is a domain and a particular domain length L of t, and the assumption is within one L of t there is a local steady state. So the dynamics for the order region is reached its own steady state behavior. That is one assumption, and the second assumption is different domains are independent. So if you take these two assumptions, you can actually show that in the coarsening phase, S of t average will grow as 1 over square root L of t, or L of t is the growing length scale that is standard here, and the variance of S is actually a constant. So it does not vary with time. So the variance constant you can understand this way, that in every domain it grows as 1 over L of t, but there are these many domains. So when you multiply and add things up, this gives you a constant. So this is remarkable that the fluctuation of the interface is constant throughout time. I mean, there are initial fluctuations, but once it is in the coarsening phase, in the scaling phase, it's constant. So the fluctuation is always there.
Orgo, you have 2 more minutes
Okay.
to wrap up. Yeah.
Yeah. So this is also verified numerically. So this is the characterization of the interface in the coarsening regime. Now the question is the characterization of domains. This is the figure we have already seen, that here the domain is struck and here there are many domains. So usually what people study is the average domain length or typical domain length. But in case of FGPO, this is not applicable because the average domain length is only order square root L, but we have macroscopic order. How do we explain that? The way to explain is to go for the maxima. So if you look at the external statistics, that is only dictating the order in the system, and that comes out with the correct size. However, it has a very interesting fallout in the coarsening phase. So for example, when we go for the time dependent behavior, we see that this is the cluster size distribution. This is a nice power law for small cluster sizes, but there is a time dependent cutoff. So as the domain grows, the cutoff also pushes through, and this is of order L of t, which is the original domain length scale. So now we know that even when we are looking at coarsening, we have to look at this tail of the distribution. That is not the typical domain size, but the maximum domain size to understand coarsening. But now when we look at the maximum domain size, there is a consequence. So you remember we have these two assumptions: the local steady state and the statistical independence of the subsystem. So locally within a domain, we expect this kind of distribution, and we have checked that this is an exponential. So now you imagine there are different subsystems. Every subsystem has a local maxima, and you are looking at the global maxima. The global maxima will be maxima of the local maxima, and you are drawing the local maxima from exponential, so the global maxima is a dumbbell. But now, what will be the average value of the global maxima? That is proportional to the domain length scale, but there is a very interesting finite size log L and log T behavior. So one expects that during coarsening, it does not care about the system size, it is only coarsening. But since in FDPO, we have to look at the maxima, the effect comes in this log L and log T dependence. And actually we have shown that if you try naive scaling, you don't actually scale. But when you take care of the logarithmic corrections, you see the scale. So the logarithmic behavior is actually the important dominant behavior in coarsening of FDPO. So this is the second important result which we try to emphasize. So I am done. So this is in summary. So we try to characterize FDPO through two quantities. One is the maximum cluster size, which is representative of order, and the interfaces, which is the representative of disorder. Secondly, we found some non-trivial positioning behavior, like the fluctuation is constant throughout in the interface, and the domains, they have a logarithmic behavior in system size and time. And finally, even though there are massive fluctuations, and one do not expect local steady state, but still local steady state is working and it can actually explain this behavior. So this is what don't get. That's it.
Okay. Thank you. Thank you, Orko. We have time for one really quick question. Yes. Could you please unmute and ask?
Is it so? Why do you see KPG scaling here, not Edward Wilkinson?
Oh no, we have done it for Edward Wilkinson also. I have not shown it here. That's all.
Okay.
Okay. There's another question, I think, or are there any other questions for Orko? If there are no more questions, let us go on to the next speaker, Urvashi Nakul. Thank you, Orko. Can I ask Urvashi to share screen?
Yes, ma'am. Can you see the slides now?
Yes. Mathematical and computational study of active Brownian particle is what Urvashi will tell us about. You can start.
Yeah. Hello, everyone. I am Urvashi Nakul from IIT Madras. This is our recent work on active Brownian particle, the theoretical and computational study. So this is the outline of my talk. So what is active Brownian particle is? From the name itself, we understand that Brownian particle means having a collision with the molecules in the medium and leading to a random motion, where active as a special case means that it consumes energy from its environment and lead to a directional motion through its self propulsion. It requires no external energy, means the force, and it is relevant to wide variety of biological and soft matter systems. For examples, in biological processes, we have seen molecular motors, bacteria like E. coli, and these are the pictures of this microscopic biological quantities. So the motivation for our work comes from this experimental paper, that is acoustic trapping of active matter. It's a Nature Communication paper in 2016, where they confined the active Brownian particle in 2 dimension using the acoustic tweezers, and where they tune the strength of this harmonic trap from weak to strong trap to understand how the distribution changes. They found that so what exactly they have found, confining the active genus particles, they found the probability distribution, which is at the y-axis, and x-axis is the scaling radial distance, that is r by u naught t tau r. U naught is the active velocity, propelling velocity of the particle, and tau r is the reorientation time of the particle. So in the A figure, we see it is for the weak trap when the trap stiffness is very low, and B is for the strong trap when the stiffness of the harmonic trap is strong. So what do we see here is at weak trap, the peak of the distribution is at trap center, that is r is equal to 0, and as the trap stiffness increases, the peaks shifts to a certain magnitude of that RC given in the paper. So this is the whole motivation for our work here, where we try to characterize the theoretical model to understand how how there is a shift as the strength of the trap stiffness increases. So our theoretical model is defined as the particle propels with the u naught constant velocity in the u cap direction, performs rotational diffusion with the diffusion coefficient dr. It reorients with angle theta due to the thermal noise. The system is present in the viscous medium given with the gamma friction coefficient and confined under harmonic potential of trap stiffness kappa. Kappa is the main important parameter over here. And we can write the overdamped Langevin equation for the active Brownian particle in the absence of translational diffusion, such as the frictional force is equal to the force due to the self-propelling particle, and minus kappa r is the opposing force due to the trap. D theta by dt is the change in the angle orientation due to the thermal noise. So this is the numerical simulations. We discretize the Langevin equation for the given step and averaged over 10 to the power 5 particles, and by discretizing we try to find out x and y coordinate as a function of time and changing into a polar coordinate, that is r and phi. Main motive is to find out the radial and angular probability distribution, that is phi of r and f of chi at given r. That chi and r I'll introduce in the later slides, and these are the parameters. Importantly, kappa is to be noticed that it is 10 to the power minus 4, which we call a strong stiffness, and 10 to the power minus 6 as a weak trap stiffness. For the given system, we can write the overdamped Langevin equation as well as the Fokker-Planck equation. The overdamped Langevin equation, which is velocity equal to the net velocity equal to v naught u cap minus kappa by gamma r. R is the position vector. Similarly, for the steady state, the whole system is the calculation has been done for the steady state, which means that the probability distribution is no longer a function of time, and writing the Fokker-Planck equation as the derivative with respect to time of p goes to 0 equal to the drift term, and in addition to the rotational diffusive term, that is the two terms given here, and j is the probability current. Right, this systematic diagram of this problem can be understood in the 2D plane, where the particle is, or the particle in the space given with the u cap as a direction, which I have told earlier, which is oriented at angle theta with the x-axis, and r and phi are the polar coordinates. So our aim is to find out the p of r u cap. We can convert it into polar coordinate system, that is p of r phi theta. And since it's a radially symmetric problem, a symmetric harmonic potential has been applied over this system. So we can consider the separation between phi and theta, that is the given parameter over here is the chi. So from the variable separation, p of r chi can be written as phi of r f of chi at given r. So these are the calculation detailed step for the for our theoretical work. So what we have done here, we have multiplied the Fokker-Planck equation with v velocity and integrating over du cap. So these are the very detailed technical steps. So mainly we can focus on the term in the box, that is integration over u cap dot grad p du cap. That took a lot of time to be calculated. So what we have done, we have changed in polar coordinate system. Sorry. And we end up finding the mean and variance in cos chi. Yeah. So mainly for finding the further steps for the calculation, we need to find out the mean and variance in cos chi. So what we have done, we have calculated this mean of cos chi theoretically using the steady state solution, and this is the result for our mean of cos chi, that is r by rm, from the theory as well as from the simulations, matches quite well, which we can see from the plot. And this is the result, theoretical result, the expression calculated from the theory also matches quite well with our simulations. So keeping all these results all together, we are able to write down the theoretical expression for the probability density, that is phi of r, and using which while multiplying with the angular strip, that is 2 pi r, we can find out the radial probability distribution. So this is our main result for the probability density, the for the radial distance, that is phi of r on the x-axis, y-axis, and x-axis is the r by rm, the scaled radial distance. These two plots are for the two different values of kappa, that is weak trap and the strong trap. So from the weak trap, we can see that simulations tells us that there is a peak at r is equal to 0, and with the theory also matches with the tail of the simulations. And we see that for the strong trap, how the peak from r is equal to 0 shifted to r is equal to rm. Similarly, we can write the radial probability distribution, which both from the simulations and the theory found to be very close to each other, and we can see how the peak shifts from the weak trap to the strong trap. So writing the mean results for the probability density phi of r, interestingly, there is a parameter alpha in red mentioned over here, that is dr minus 3 kappa by gamma. This parameter tells us that when it is positive, it indicates the weak trap, and the peak is at r is equal to 0, which is near the trap center. And as changes the sign, found to be negative, it represents the strong trap, and the peak is away from the trap center, that is around rm. So with the increase of the trap stiffness, we are able to observe the transition from passive to active behavior. So this is the angular probability distribution, completely the simulation results. So interestingly, this tells us how the orientational picture of the active Brownian particle. So from the distribution, as we remembered that there is a peak at Rm, so means the particle is accumulated at the angular region away from the trap center. So we have explored the orientation angle for those particles. Considering the angular region, we have we have considered these different values of r representing with the different colors. For a lower value of r,
Urvashi, you can finish your talk very quickly and then we can go to the question.
Okay, ma'am. Okay.
Yeah.
So at lower value of r, we can see there are two peaks in chi. The distribution shows maxima at around chi is equal to 30°. And as the r increases, we can see that these two peaks merges together, and at maximum near the rm, there is a peak at chi is equal to 0, which tells us that for lower values of r, particles are moving in either direction, clockwise or anti-clockwise. And in the outer ring, there is a peak at chi is equal to 0, indicating that the orientation of the particle u cap directed along the radial direction r cap.
Urvashi, I think you have to conclude right now. There are lots of questions.
Okay, ma'am. So
And
the
we don't have
conclusion,
time, yeah.
yeah. So conclusion is we found the radial probability distribution
Sure.
with the trap stiffness increasing. There is a shift from the passive to active behavior, and from the angular probability distribution, we understand
Right.
the orientational profile of the particles. Thank you.
Okay. Thanks, Urvashi. So PK has a question in the chat box. Is there a scale that distinguishes weak from a strong trap? What sets the scale? So basically, I think what that means is, how is that RM? I mean, how does that depend on the trap stiffness? I think that is that correct, PK? Is that what?
Yes, yes, that's what I
Okay.
was.
Yeah, so RM is actually V naught. Yeah, so RM is gamma U naught by kappa. It is inversely proportional to the
Okay,
trap
okay,
stiffness.
okay.
Okay. So in the experiment, did they do the experiment at several stiffnesses, or just one weak and one strong?
Yeah, so yeah, they have presented these two different values. Means these
Okay.
are like scaled parameters.
Sure.
So yeah, exactly. I cannot tell which which kappa value because they have the scaled kappa stiffness.
Okay. I think Urna had a question.
Yes, hi.
Yes.
Yeah.
Urvashi, so you showed this phi r that you have found, the
Yes.
radial distribution. So in the weak strap limit, this should actually go to the Gaussian. So does it go? It's not very obvious from the expression.
From the experiment or from my
No, no.
results?
Theoretically, it should go to the Gaussian in the weak trap limit. So your
Yes.
expression, does it go?
Yeah.
Ma'am, what we have understood, we have considered a single approximation. Maybe due to that, it does not shows up exact peak at r is equal to 0.
Yeah,
That
what
is
is
what.
that approximation that I missed?
Yeah, yeah. So the approximation is while calculating this variance in cos chi. What we have done, there is a expression over finding the mean of cos cube chi. So we have neglected the higher order term while having the expansion over mean.
Okay.
So yeah, that was the approximation.
Okay, thank you.
Okay. Thanks, Urvashi. There are other questions, but I think we have to move on to our next speaker, Dr. Mamta Sahu.
Okay, yeah. Thank you, ma'am.
Yeah. Okay.
Hi, thank you. Let me
You have to
first
share your screen.
share
Yeah.
the screen.
Yeah.
Okay, so now it's visible?
Not yet.
Not yet, okay. I see.
Yes. Now it will be. Yes, now it's visible. Okay, you can start. Inertial active Ornstein-Uhlenbeck particle in a magnetic field.
Hi, let me first thank the organizers for giving me this opportunity to be part of this conference as well as to speak about my recent work. So I'll be talking about, I think so far we have discussed, we have already heard a lots of talk based on the active matter. So today here I will just talk about inertial active Ornstein-Uhlenbeck particle under the action of magnetic field. So as already we have heard, this active matter is a systems where especially this is they are inherently driven far away from equilibrium, and the constant active matter self-propelling by them and generate the system. Examples of systems, you can just have flow.
Is there I think there's a problem with audio. Is it?
Issues inside.
Yes.
Yes.
So
Okay.
actually, there are some standard AUP model which for the constituents in such matter systems. But okay, is it audio problem? So I think everything gone.
No, now it is suddenly much better. Now the audio is suddenly much better.
Hello. Oh, okay.
Yeah, can you hear
so
me? Yeah, now
yeah,
I can hear you clearly. Before I could not.
Okay, I see.
Maybe
so now, but the slide is visible.
Sorry.
The slide is visible.
Yes, it is now.
Okay.
Yeah. You can start from where you left. There are about 8 minutes, 9 minutes.
Examples of active.
Okay, there is a problem with audio again. Can you maybe just start from where you left off the last time?
Okay, I see. Is it okay now? Can you hear me?
Yeah, now I can. Now I can. Yeah, maybe we can start from here.
So there is a delay. Maybe this is, I have included some videos, so that is making me problem maybe. Okay, so actually,
oh, it's completely gone.
Yeah. So what do we do?
already we discussed that there are process.
Mamta, you are not audible at all.
Is it possible to call her? I'm not sure she's even aware of the fact that we cannot hear her.
So should we start the next talk and then we can
maybe let's do that while she kind of if Tripta is ready, we can start.
Okay.
Huh? Okay. Thanks,
so,
Mamta.
Yeah.
We'll get you back. So if you will stop sharing the screen, Mamta, then we can ask Tripta to start her presentation now about 7 or 8 minutes early. Tripta, if you are ready.
Yeah, am I audible?
Thanks.
Yes, you are audible, Tripta, but yeah, you seem to be audible.
Ranjini, should I share or wait?
No, you
Yes,
can.
please. Yes, please. So Tripta, you have 30 minutes, so maybe around 23 minutes after you start, I will just switch on my video and audio, and then yeah, so that we can at least have 5 minutes for questioning. Yeah.
Yeah, Ranjini, maybe you just tell me, because when I do the full screen, I cannot see.
Sure. Maybe I'll just alert you 20 minutes after your talk. So yeah, so that you can finish in the next 5 minutes, and we can go on to questions. Okay,
yeah,
thanks.
That's really nice.
Life as a matter of fact.
Okay, hello everyone. So I'm Tripta. I'm working at ISR Mohali, just joined last year. So I will be talking about a topic which is life as a matter of fact. So what do I mean? I will hope I will be able to convince you about the work that I did, that the life is actually a matter of fact. So the outline of the talk is that I'll introduce you to matter. What do I mean by matter, fat, life, and life transition, matter to life transition, and so on. Then today, I think I will focus on fluctuations and biomembrane curvature, tension, and then there will be some outlook and some ongoing and future problems that I plan to work on here in the lab. So these are some of my publications that I will be using. I just listed. So among this, the first one is one that I have Aysar Mohali affiliation with my collaborators, but all others are my postdoc publications. So matter to life transitions, what do I mean by that is that if we really go down to the building blocks of what matter is made up of, so it is sugar, amino acid, nucleotides, and fatty acids or fats. So I have been working with fats or fatty acids, and these are building blocks, and they form macromolecules
Hi, Mamata. Yeah.
such as polysaccharides, protein, polynucleotides, and loose macromolecule assemblies. So the question often synthetic biologists ask is, what is this transition from matter to life? How does this life that we see, how does this transition happens? So there are various ways to address this problem by doing research. One is bottom up, where you start from the matter and try to build up the life. Other is the top down approach, where you take, for example, tissues or organism or cells, and then you go down and see how the whole complex system works when you bring it down to the matter, to the building blocks. And one such example is also like one can work with the cells, and one can also often form compartment out of the cell, which is less complicated compared to the cell. So this compartment that I show you, green and red, this is called giant plasma membrane vesicles. So these are basically the compartment which are derived from the cell. It contains everything in the cell except the cytoskeleton. So this is often used to study the properties of the cell without cytoskeleton. And this is an interdisciplinary field. So like I said, the different kingdoms of life have different in experience and functionings, but if we go down to what they are made up of, we can often quantify that into very concrete branches. And the title of my talk is actually from this book of one of my collaborators. So all these macromolecular assemblies and building blocks that I introduce, they fall into a category soft matter system. And Ranjani Dee is also a soft matter experimentalist, one of the first I met during my PhD. So biological functions of soft materials are controlled by basic physical principles. That's why I'm a biophysicist, so that's why we can study the functions using the physical principles. So why they are soft? Because it's easy to deform them, and thermal energy is the energy scale. The response is non-linear, and it's very large. Shear modulus is very small compared to solids. So for solids, it's very, very high. And then one example of such soft material is, for example, biomembranes, cellular membranes, which is quite complicated, but we can either study the whole membrane, which is a fluid membrane. So this kind of membrane, which is in 2D, and if we ignore the thickness compared to the span of it, which is in microns, it's kind of two-dimensional fluid. It's also called complex fluid because it's composed of a lot of molecules embedded in it, and they are all there is no bonding between these molecules. The other example is virus, for example, coronavirus, which is an example of a colloids. Then all biopolymers, biosurfactants, and today I will be mostly talking about biosurfactants or lipids that form cellular membranes. So as a physicist, how do I study the physical property of submembrane? So one thing is the thickness. The other thing is like in equilibrium, there are interactions, and they all balance and then give rise to certain area per molecule and thickness of the bilayer, which can be changed if I bend the membrane or if I stretch the membrane. So there is an energy cost associated when I want to change the membrane conformation from the equilibrium conformation, and it costs energy. So one of the problem that I I'm interested in to look at the fluctuations of these membranes, which are nothing but the Brownian motion. So there were a lot of talks yesterday and also before and also today about active matter. So how does fluctuations can be active? It can be active if so. I should show you this movie, which is I hope to play, which is, yeah, so it's playing now. So this is a compartment that we make in the lab, like a cell, and you see these fluctuations, and these fluctuations are actually the fluctuations because the solvent molecules are kicking on the membrane, and of course lipids are kicking back, and that's how these are the Brownian motion, thermal Brownian motion. In equilibrium. So there is no active processes going on. There are no active proteins or something. It's just a self-assembly of lipid molecules, closed compartment, and we can see these fluctuations due to this Brownian motion. So then what can we get out of it? We can look at it. We can look at the by using the Mung gauge, we can find out what is these fluctuations. And then from the fluctuations we can find out the bending rigidity, like how easy and or difficult to bend such membranes. So for example, it's the material property. So in this formula where I calculate the correlation between U and U, so it's a mean square amplitude. If I calculate, that depends on kappa, which is the bending rigidity, and this is in spherical harmonics in the Fourier in the Fourier domain. I can, if I plot these average over this quantity which is along the y-axis, which is actually related to the autocorrelation function of the mean square displacement, then one can see how it depends on different modes when we fit these fluctuations. So we can observe these fluctuations in real time, like I showed this, I played this movie to you. And then just by looking at the mean square fluctuations, we can calculate the bending modulus of the membrane. Then what happened that when we have proteins sitting in these membranes? So I told you, like in biological membranes, there are a lot of things like proteins and a lot of active processes going on in the membrane. And then if there is a transmembrane protein or any kind of active process that's going on in the membrane, that also has an effect on the membrane fluctuations. And we can also measure that, and it's a very controlled system in the sense that we can just look the active and passive state of the same GUV by adding the ATP that is required for the activity of the protein. For example, I worked with a protein called sodium potassium ATPase, which is a transmembrane protein. So we have reconstituted that protein in the membrane, and then we could add the ATP and look at the active and passive fluctuations. So this was a chapter which was published, and then we could also model it just by using the equilibrium relaxation dynamics. We can measure the relaxation time in the presence and absence of the activity. So this is an example, simplest example for me for the when I wanted to learn and do experiment in the lab. This was one of the first example for me to learn about Brownian motion in the active and passive state and how to calculate the relaxation dynamics for the equilibrium and non-equilibrium system. And this was another publication by Helen, which showed that if there is a pump sitting, then if I change the membrane composition, then the kappa, which is the bending rigidity, it depends on the membrane composition, which is not surprising because these fluctuations requires membrane bending, and how easy to bend it obviously will depend on its composition. So the kappa will obviously depend on the composition, and it will change with the lipid. It also changes with if the membrane is now having an active protein or not. As you can see that with the activity, where this second column it says ATP, the bending rigidity is actually it's reduced compared to when the system is in the inactive state. And also if there is no pump in the membrane, then also you get little bit different kappa. So it seems like once we have this transmembrane protein sitting in the membrane, presence of the protein itself can change the material property, as well as its activity can modify the fluctuations. And she could actually extract the turnover time of the protein by, so what she did, she just used this relaxation dynamics, and then she looked at how the correlations in decay in time for a given mode number, and then she could find that there were two kind of relaxation dynamics. One which was same as when it is an inactive system, and the other was when the system was active. She found two relaxation time. One was the usual equilibrium relaxation time, which is Milner-Safran relaxation dynamics, and the other was related to the turnover time of the protein, which was independent of the mode number. So it actually gives a lot of the fluctuations gives a lot of information about what is going on at the small scale. And the fluctuations that we look in the microscopes are actually fluctuations of the microscopic object like a cell or something. Now, once we modify the composition of the membrane, I showed you that, it will modify the bending property, material property of the membrane. We can also modify the material property just by making a kind of environment having a bilayer with two different kind of leaflets. So one leaflet having molecules which are absent in the other leaflet, and this immediately gives a kind of leaflet asymmetry, and this gives rise to curvature, which we call spontaneous curvature. So by doing that immediately, by creating such kind of frustration or such kind of, by embedding such kind of molecule, we create an asymmetry, and that give rise to a kind of tension in the membrane, which we call also the spontaneous tension, and the corresponding curvature is the spontaneous curvature. And when we have such kind of curvature generated, we often see generation of tubules. So for example, I showed you here some examples which are published where the first case, A is same as D, where we see a lot of tubulation just because we make the leaflet asymmetric. In the E, we have again a leaflet asymmetry where we have now a protein sitting on one of the leaflet and not on the second one. And the F is an example where we have membrane domains. So membrane has a lateral heterogeneity and not a transverse heterogeneity like in case of leaflet asymmetry. Then also we get a lot of shape transformations. So a lot of material properties can be controlled in the lab by tuning what is the composition of the membrane, and also by changing the composition inside of the one leaflet compared to the other. Then we could also see that, even if we don't change the leaflet asymmetry, we can, if we just change the solution. And so what I mean is that one leaflet is in contact with one kind of solution, and the other leaflet is contact with other kind of solution. Then also we create these shape transformations, and all these are when these transformations are present, then it also affect membrane fluctuations, because these fluctuations can exist in the absence of these spontaneous curvature. But once these such a spontaneous budding or tubulation occurs, it also affects membrane fluctuation. So this was a case where we also showed that, if we have a GUV or a compartment with two birds, they can also have a stochastic movement of the birds along the membrane. So it's a single membrane. So the two birds can be like a Mickey Mouse kind of conformation, or it could be like a linear necklace conformation, and it can go from one state to other stochastically. So we found these kind of transformations, and then we went on, because it's a fluid system, so it's easier for them to just glide along in the plane of the membrane. And then we can have a whole phase diagram, where it depends where we start and what we get by having such transformation which are created by introducing asymmetry in the environment. So it of course affect fluctuations, but it also stabilize regions spontaneously because of spontaneous tension. So then the next step, what we tried to do is we checked if we could also create fission of these two separate compartments, and we could actually induce fission by increasing the asymmetry. So there is a neck which connects the two compartments, and the neck can be cleaved by increasing the asymmetry. So here we plot spontaneous curvature versus the GFP coverage. So GFP is the protein which is sitting on one of the leaflet, and just by increasing the coverage, we could induce the asymmetry, which is the physical parameter corresponding to that is spontaneous curvature, and we could induce fission of the compartment, single compartment, into two. So we could tune it, we could control it, we could also measure the force, which is now proportional to the material parameter kappa and spontaneous curvature, and also the neck curvature. Now we get a lot of shapes, different kinds of shapes, and these are all single membranes which are forming all these kind of shapes. And as I said, there is a transformation. So I have this phase diagram. Okay, maybe I did not show it. So there is a phase diagram, where you can actually go from one conformation to other. So for example, like I showed for A and B, that you can go from A to B, these buds can just glide. One can go from C, one can go from, for example, from E to another conformation for E. The F can also be different conformation. These buds can just glide, and so on and so forth. So this is all important because, when we study active matter, or when we want to study active fluctuations or active systems in the lab, and we are making such systems in the lab, then it's important to know what the asymmetry is doing to such systems, because it's just equilibrium systems are enough to give rise to such instabilities. Can form multiple bud, can induce compartment division, can induce tubulation, can form different kinds of shape transformations without any activity. So the activity, when we add activity on in the presence of such system, the activity does something on top of what we already have it in equilibrium. So this was another problem that we worked on, that how do we measure such tension and in the presence of such spontaneously generated shapes, stabilized by spontaneous curvature. So of course it affects the entropic undulations. So how do we measure it? So one way to measure is actually to use this macromechanics, where we actually hold the compartment and then aspirate it, and then it's a single compartment. So then all these tubules, they kind of just flow into the mother GUV, into the pipette, and then when we calculate how much area is stored in this rugged or all these surfaces with so much of extra area, then that also give you an estimate of the tension, because when you apply tension, you can get rid of these surfaces, these fluctuations. So that in one sense, if you measure how much area is stored, you can also measure the tension. And we also found that, if we form these compartment directly from the cell, not like we make using the lipid in the lab, but directly from the cell, like the one I showed you, GPMVs, then we also found that, if we now plot tensions versus the relative area change, these all will have different slopes, which means that, when we have a compartment derived from the cell, and when we have a cell, and when we have a compartment which is derived from another cell with and without these shape undulation, they have a very different mechanical behavior, because the slope here is quite different for the GPMP with and without tubules. And this is how we measure it. So we apply a suction pressure, and then we measure the corresponding so we know delta P, because I'm the one who is changing delta P, and then I know the structure of my system, and then I plot this kind, I make this kind of plot, and from the slope gives me an estimate of the spontaneous curvature, which is sigma. So we did it for different, we tried it for different asymmetry of the glycolipid, and M is the spontaneous curvature, and then we can measure it by measuring this sigma. And this also gives us an estimate of the thickness of these tubules, which are stabilized by the spontaneous tension. So in general, we can have all these kind of different shapes, fluctuation, tubulation, stochastic movement of the buds along the fluid interface, compartmentalization, budding, and there is no activity in this system so far. But we can, as I showed you, like we did measure bending modulus of the membrane in the presence of activity, and we found that it is reduced in the presence of activity. So we also plan to now do more in this direction in the lab. And so these are some of the examples that I gave you. And this is my team at the moment. So if I just say the name, it's so Jay, Sanat, Mayur, Utsav, Abhimanyu, Syar, Harshit, Suhasi, Smriti, Madhurima, and myself. So they are just students in my lab. And then I thank Professor Cornelius for giving me an opportunity to collaborate with him. I have a Ramalinga Swami re-entry grant with him. And then Abhishek Chaudhary is having a joint student, PhD student with me, Abhimanyu. We are both mentor for the student. And then John is one of my other collaborator in the lab. So I thank all of these. And so all these systems that I spoke to you about, I mainly spoke about biomembranes, but there are a lot of examples of soft material. And
so Tripta, you have about 4 minutes left.
Okay, sure.
Yeah.
So it's important because we are made up of soft material, and so it is possible to measure the micromechanics by doing micromechanical experiment. It's possible to measure the mechanical properties such as bending modulus, stretching modulus, and we can also characterize active processes apart from the passive processes. So if we have such a compartment, for example, this one, if we have a compartment like this, we can actually conduct both the active and passive experiment on the same compartment, which is not possible in case of a cell, which is quite complicated. So I think I will just stop here, and I want to thank Ranjini Di, and also I want to thank the organizers for this opportunity, for giving me this opportunity. So thank you so much.
Thanks, Tripta. We have time for many questions.
Maybe there are no questions.
Okay, so till the time somebody thinks of some other question. So see, you showed some temporal autocorrelation function of inactive as well as active membranes.
Yes.
Towards the beginning. So
Yes.
The active case had two different relaxation times. So I was
Yes.
just wondering what kind of processes those two timescales correspond to. What is the order of these timescales? What are the order of the timescales?
Yeah. So one time scale was like if I don't have any activity in the system, for example, just I take a membrane and I just measure the correlation of height fluctuations with time, how it decays with time. Then I get one kind of relaxation.
Right.
And that just relates to how the fluctuations are correlated in time. And the other time scale was related to, which is, as you can say, so the first relaxation time depends on the mode number, like the bottom figure, if you look at the left one. So first relaxation time, and the x-axis is the mode number. So it depends on the mode number. But the second one, which is about half a second, as you can see, there is a red dotted line. So this we get almost constant independent of the mode number. And this was something related to the transmembrane protein that we have put in inside the membrane. So I'll just show you what exactly we have it in the membrane. So we have a protein sitting in the membrane, which is transmembrane protein, and this protein pumps sodium and potassium ion across the membrane when it is active. So it has a turnover time, which means that how much time it takes to complete one cycle. So 3 sodium and 2 potassium. So that's the turnover time. And this half a second is the turnover time of the protein in the membrane, which is independent of the mode number. So it's quite stable, and we could extract that just by looking at the active fluctuations is what was the result. So this is not my result. This is published by Helen. But what we tried to do, which is unpublished at the moment, is we tried to reconstitute the same protein in raft membranes and more complicated membranes, because this is a simple membrane, a DOPC membrane. And then we want to see if there is any change in the turnover time scale of the pump because we changed the membrane environment. So that is still unpublished, but we could extract turnover time of the protein just by looking at the fluctuations.
Thanks. So there's a Department of Physical Science seminar has raised a hand. I think that's PK.
So so you have this active and inactive timescales, whatever you saw here. Yeah. In active case, it looks like there are 2 exponentials. What are these timescales which are effectively there are 2 timescales? What are these 2 timescales? Like I can fit it into 2 exponentials. Look like
we actually fit 2 exponentials. So one was yes.
What are they correspond to?
Yeah, so one corresponds to the equilibrium relaxation time, which is the Milner-Safran relaxation time. So this one.
Okay.
So if I write this Langeau equation and if I just calculate this autocorrelation, and this is the Milner-Safran relaxation time, which depends on 1 by L cube. It depends on the mode number. So this is equilibrium fluctuations. And the second one is when we have a protein and there is a turnover time cycle and the time corresponding to that, which in our conditions was tuned to half a second. And this is the second relaxation time. Actually, it's independent of the mode number and it's correspond to the turnover time of the protein.
Yeah, okay. Thank you.
There is another question. The videos you showed are two-dimensional, but in reality, fluctuations are happening in 3 dimensions. So
Yes.
how do you take care of this?
Yes, it's true how to take care of this, but the thing is like that's why we are fitting spherical harmonics to the GOVs, and because we have spherical GOVs, and so there are different way people extract properties. One is like Fourier decomposition. As you correctly said that it's a 3-dimensional system, so we actually fit spherical harmonics to the GOV to extract
but the data has this 3-dimensional information, or it's just a video and right, do you have a 3-dimensional information?
Yeah. We can have, it's yes. So we can have so when we take a phase contrast microscopy image, we have information only about 2D, which is the equatorial plane. But if we take a confocal image, then we have information about the 3 dimensions. But this movie that I showed here, it doesn't have information about the 2-dimension. It's just sorry, about the 3-dimension. It's in at equatorial plane. What we see.
Yeah. Okay. Yes, that was it. Yeah, okay.
Yes.
Are there any other questions? If there are no questions, maybe we can thank Tripta.
Yeah. Thank you so much.
Yeah, thanks a lot. And we can move on to Joydev Chakraborty.
Okay, so let me first share the slide and make sure that you can see that. Can you hear me though?
Hear you very well.
Oh, very well. Okay. Oh, hold on a minute.
Sure.
Hello? Hello? So can you see my screen?
Yes. It's gone. Yeah, it's back again. Okay. So I'll remind you, Joydev, after 20 minutes, and then please try to finish in the next 5 minutes, and then we'll go on to the questions.
Okay, so yeah, I'll try my best to do that. I could even finish before that.
Okay.
So let me first thank these organizers for giving me an opportunity to share some of my research, which is done in our group. So I belong to the Department of Chemical, Biology and Mathematical Sciences, Desenbo Center, Kolkata. The work that I shall present was primarily done by senior graduate student Rahul Karmakar. And I also would like to acknowledge Oishani Ghoshal, who did her PhD in IISc, and on her way to Israel for postdoc, she spent some time in our group, and she initiated a part of the work actually to begin with. Okay, now as a background, or the motivation of the work really comes from the recent pandemic that we are undergoing. One of the things that has come like enforced upon us is use of mask. Now, the reason for using mask is clear, that we like to stop the airborne respiratory microdroplets. So basically what happens when someone speaks, sneezes, and for almost any activity, we emit microdroplets, which are called respiratory droplets. For an ailing person, such droplets actually contain virus and bacteria and microorganisms, which actually can infect the other healthy subject. Now, mask is recommended by the WHO as a primary, let's say, measure to intercept the respiratory droplets and to prevent the airborne transmission of the disease. Now, if you look at the mask, typically a mask material which is responsible for stopping the microdroplets are all fibrous, essentially polymeric fiber is a porous medium through which some particles can go and some particles cannot go. So it's like a semi-permeable medium. Now the question is, what properties of those fiber which actually helps to stop the airborne microdroplets, which is the purpose anyway. Now you can immediately see that the problem that we are talking about is passage of particles through a polymeric network, which is definitely pretty much studied. People know a lot about how particles actually can propagate through this kind of network. Now, if you consider the physics of mask, there are two things that you have to keep in mind. First, if you look at the WHO guideline, which is pretty much there in many places, what they recommend is a multi-layer mask, where the outer layer which faces the air side consists of hydrophobic material. The inner layer which faces our mouth is typically made of hydrophilic material. In between, typical mask material that is used are hydrophobic. So you can think of the mask as some material with a middle layer, which is certainly the important one, where all the interception happens, is something confined between two layers. One is hydrophobic and the other is hydrophilic. So this is one point that one has to consider if you want to understand the physics of mask, like how the particle is intercepted in a mask. Second point is also important, with the breathability. Like, you can immediately see that when we inhale or exhale, we basically force, air, when the air gushes either inside our mouth or goes out of the mouth. So whatever thing that takes place through the mask should be able to withstand the gushing air. Now, there are many experiments done in recent times where it shows typical pressure that acts on the outer surface of the mask, which faces the air side, is about 2 Pascal. And so that's the kind of pressure that acts on the mask. So you have to consider that whatever things that is wrong through the mask network is subject to this force. So inherently, this system is non-equilibrium, and one needs to consider the steady state condition that comes out. So this is what we try to understand, given the basic, let's say, kind of knowledge that we have from the WHO guideline, and the fact that we have to understand, we have to consider the air pressure that is acting due to breathing, which could be either exhalation or inhalation. The kind of thing that we are considering here is inhalation. So it's a kind of a model, it's a very simple model. I must say that in our model, the hydrophobic, hydrophilic are taken in terms of certain interaction parameter. We have not considered the full, let's say, details of those interactions. We just try to model it in as simple way as possible. So it's a caricature that is shown here. I hope my cursor is visible, right? My movement on the cursor. The one that's shown in red is the one which faces air, and the green one faces, our mouth, which is typically having like water, saliva, etc., etc. Now, the one which is facing the air side, we call it H wall, and the one which faces the water side, inside the mouth, we call it P wall. Now, hydrophobic material, let us say that the the material that we consider is having, let's say, some, two kinds of, bits. Of course, we are using simple bit string model of the strands, and there are, identical strands like this in the material. Now, some bits are called H. So these are the bits which actually responsible for cross-linking between the polymeric strands, because in the end, it should form some kind of, network where there would be cross-linked, bits. And the P bits are one, which we call so-called polar bit, if you want. Now, these bits are having interaction which is less effective compared to the interaction between the H bits. Now, so far the wall is concerned, the wall which faces the air side, which we call as H wall, they like H bit. So they have preferential interaction with the H bits. And the wall which is inside the wall, inside the mouth, towards the mouth side, which is facing water, saliva, etc., they prefer the P bits. So these are the simple kind of interactions that we consider in the system. Now, in this slide, what I tell you is essentially what I already told you, but slightly in terms of formally. So we have, so far as the polymer bits are concerned, which are forming the network, there are non-bonded interaction, which essentially like saying that some of the bid like each other more than the other bid. So that's what is reflected here, that epsilon HH is larger than epsilon PP. On top of, there is a bonded interaction, which is essentially like bond between different polymeric bits. And this could be both P and H bit, and we don't distinguish between H and P bit so far as the bonded interaction is concerned. So bonded interaction could be two types. One is stretching, and the other is angle, which is essentially like saying that if you want to bend the polymer, the strand, it will cost some energy. Now, in addition to all this interaction, material, we have tracer particle. Now, tracer particle, if I go back to the earlier slide, the tracer particles are taken to be on the H side. Now, the tracer particles are taken to be having more favorable interaction with the P bits. Okay, so that essentially will mean that if you want to use the word polar, then that will mean that the tracer particles will prefer to interact with the polar bits and the polar side of the wall. And that is because the kind of system that we have in mind as tracer are the micro droplets, which are having water as in content. And we know that water is a polar medium. So keeping that in mind, we are taking interaction of this kind. Now, of course, we have to consider the interaction between the bead and wall. Now, you have to it's clear from the setup that the bead wall interactions are asymmetric. One wall has got, we take again a very simple interaction like this, which is pretty standard in literature. The H beads prefer the wall H, P beads are preferred by wall P, and tracers are having more favorable interaction with, again, P wall than the H wall. The analysis is done in a very simple fashion. We just write down the equation of motion of the bead particle and the tracer particle, and we just numerically solve this using, this is done essentially using the LAMMPS package. And these are the equation of motion, which includes the inertial terms, the friction term. Friction comes from the network itself. We just model this number, and then the potential that could be due to different interactions that I talked about. And there's a thermal noise. Similarly, if you take the tracer particle, everything is very similar, except you have an extra force which is acting on the tracer particle, because we know that there's a force which is acting due to gushing of air. In this case, we just think in terms of inside, like inhalation that we're talking about. Now, the steps of calculation are pretty straightforward. First, we prepare the entangled network. Well, I should not use the word membrane here, it's a real network. Within the asymmetric confinement, here the wall is taken to be structureless for simplicity, to keep the entire effort within simplistic calculations. And then what we do, we add tracer particles close to the H wall. Then we assume there's and we take a force which is acting from H to P wall. The tracer particle movement is tracked. Of course, the bead particles are also moving. They have motion. And what we are interested, how the tracer particle is moving inside So basically, you can think of the tracer particles are making permeation through the network till the polar wall. The question is how many of them actually can reach the polar wall? What are the interaction parameters which control this movement? So, a little bit into numbers. So we take a very simple thing. We take all the bits are like micrometer. The droplet that we consider is also a micrometer. It is typically, microdroplets that we are talking about is about, between 1 to 5 micrometers, the typical size. And then the mass of the droplet is taken to be that of water, of 1 micrometer droplet with about a picogram. And typical time for the movement, which is given by the so-called Lennard-Jones time scale, is about 0.35 millisecond. So here is a caricature of the system that we are talking about. So the one which is shown by the dashed, boundary are the two walls. One wall, the green arrow is the one which is so-called P wall, which prefers the green particles, which are the polar ones. And then the red one is the hydrophobic wall, so-called, or the non-polar wall, which prefers the red particles. Now, first thing, what we try, first of all, we take, the ratio. Okay, there are several parameters in the system. We change the ratio of the hydrophobic versus polar bead. We also tune the interaction of the polar bead with the tracer particle and so forth. And I'll tell you, not all the results, but some of the results which could be interesting to look at. First thing that we try to see, the inter-strand bead correlation function. So the idea is that you take a bead of one strand, you calculate the distance with the beads of the other strand, and then calculate the so-called correlation function, which is normal way of looking at liquid-liquid correlation. This is exactly similar calculation. Now you can see that there's a very strong peak at distance 1. So that essentially means that the strands are sort of entangled with each other, and this is sort of more confirmed if you look at the correlation function in a similar way, but decomposing into the species. Clearly the H correlation is much stronger than PP correlation, which is something that we is essentially would expect out of the construction. But what is important that we have now, this sort of ensures that in equilibrium you have a network through which you would like to see the motion of the tracer particles. We still don't switch on any force. We just no force and equilibrium case. So what happens in a natural course of motion, the tracer particles will move inside, and this is a typical. For given set of parameters, this is a typical distribution of the tracer particles within the let's say Z is the direction along the confinement. So there will be peaks. We can see peaks where the peak is like this, and then what you can think of, you can integrate around this density profile to calculate different quantities. What are the quantities that is interesting to us? If you take the peak close to this is a polar wall, the green was a polar wall, the peak close to the polar wall, then that will give me a permeation at the polar wall. And of course, we like to see the permeation should be as low as possible. And then efficiency is 100 minus P. Now, if you take, your tracer particle extremely large, then of course nothing will permeate. But that is not the case in reality. In reality, the pore size, then the size of the polymer beads, and then size of the droplets, they are almost in a similar range, and that's what makes your life complicated. So first, what we try to look at what point the permeability at the polar wall really falls to a permissible regime, which we take about 5%, below 5%. So you see that if you look at this plot, you can immediately see that the permeability falls below 5%, is that the sigma tracer, where the size of the same tracer part is about 1.8. Here again, everything is used in the unit of the bead size, which is about a micron. So about 2 micron is the kind of a cutoff, and then of course we shall be interested in tracer size, which is below this cutoff. So obviously that means below 1.8 or let's say 2 micron. Next, what we do, we come to the actual situation, we apply force. Now we take a very simple, force profile. We assume that the force is acting on the tracer particles, and then the force has got a simple linear form like this. And the way that the parameters are fixed, like, if, F0 is essentially the force that is acting at the non-polar wall. And this F0 is set in such a way that the corresponding pressure difference is around 1 Pascal, which is the experimental number. First thing what we see, that in the presence of the force, which is the force values are written here, what is the time that it takes a system to get a steady profile? Particularly, what we show here, the permeability at the polar end. And you can see that with time, they come to a steady value. And the 50% steady value is taken to be a time, which is, let's say, you can say is a saturation time, that the time the particle actually gets into the polar end. The permeability happens at the polar end. And then you can immediately see that with the force, the time of saturation actually decreases, which is something to expect. The force will help the particles to go to the other side. On the other hand, this time again increases linearly with the tracer size. Again, something you'd expect, because as you increase the polar size, tracer size, less and less particle will reach the other end. Now, the interesting thing is that with the parameter that we have taken, and if you look at the saturation time, we get something like 1.7 second, which is comparable to breathing time scale, which is about 2.5 second, because you have to remember that we breathe 12 times a minute typically. So that corresponds to about 2.5 seconds. So typical time within this model is not too bad, let's say. Now what we do, we try to calculate the efficiency as a function of the tracer size for different force that we apply, and we see a linear profile. So the force, actually the efficiency increases with tracer size, and for a given force, and again, as a function of tracer size, again there's a linear fall with efficiency with force. So this is the first thing that we sort of find about the system, that the efficiency, how the efficiency changes with the gushing force as well as tracer size, and this is a linear dependence. Next, what we do, we take different ratio of H:P, and what we vary is the interaction of the tracer particle with the polar beads with respect to that with the hydrophobic beads.
And 5 minutes, yeah? 5 minutes.
5 minutes, okay. So what we find that for 50-50 case, for example, there are two regimes of dependence. So is for 30-70 case, the two regimes. Now 30-70 means, and 50, and then if you take 70-30, when it's a poor hydrophobic rich system, the sensitivity is not really that much. So next thing that we try to characterize in the system, what is the effect of, what is the kind of motion that is taking place? In order to see that, for a given tracer size and for different forces, we find how the permeability at the polar end is changing with temperature. And interestingly, we find a beautiful RNase behavior. So there is a kind of a activated process. And the activation barrier actually falls linearly with force. So the activation, this is an activated process, and the activation barrier changes with temperature, and it's a linear dependence on the force that is acting on the system. In order to understand what is happening, what we try to see here, the energy profile of the tracer particles inside the membrane, inside the network. And then, so this is essentially, if you take different slice of along Z, you know the number of particles, you calculate the interaction, divide by number of particles, so this is what you find. Now, what is clear from the graph that, the black one is without any force. So then you see that there is there are several minima that is present, and you have to, there's a barrier. So you need an activation, which is something that we find. But as you increase the force, particularly for a large force, you see that the barrier, the barrier almost disappears, and the profile becomes quite flat. And so this is essentially what we find, that as you increase the force, then the barrier actually, the particle motion becomes easier. So that's something we already find. And the fact that the barrier falls with force is something that you would expect from this kind of behavior. So to summarize, what we find that mixing polar material with higher affinity for the droplet is the one which can give a better efficiency. We also can consider the optimum composition of the material is about 50-50 instead of pure polar or pure hydrophobic, because you can see that in the presence of the polar material, you see that the efficiency is more, and also in the presence of polar material, the tunability of efficiency is better. So that is so basically we would say that this is a much better situation than having a pure material. And then we also find there's an activated permeation process, and the barrier height can be tuned in many different ways. We already shown the effect of temperature, force. We can actually look into other effect as well, which we already have looked into. And in fact, right now, this is an ongoing work, so I cannot tell you the last word. We are now trying to look at how the rigidity of the mask material. See, so far what we have tuned are the non-bonded interactions, like the van der Waals interaction, which is essentially the physical interaction. But of course, there is a bonding interaction also. We have not yet looked into the how the bonding interaction actually affects the entire scenario. This is something that we are looking at right now. And of course, it is important to know the in more details of the activation process that you're talking about. So thank you for your attention. And I also thank the organizer once again for giving me this opportunity.
Thank you, Joydev. Questions? Yeah, is there a question? I can ask a quick question. So Joydev, N95 masks, which are made out of fibers, these are almost these are sometimes electrocharged. So can you put charge in your model?
See, in principle, yes. But the reason we didn't put charge is the following. See, if you have the droplets, we're not thinking in terms of virus. We're thinking in terms of droplet. Large droplets are not really so much charged. So the steric interaction will be more important.
It's for the okay. Yeah, okay, fine.
So that's why we hadn't, in principle, of course, you can take charge, but then your life is more complicated. You have to consider the long-range interaction, etc., which you didn't want to get into. But in principle, of course, one can consider charges.
Okay. Okay. So the efficiencies will be different, when, in the other direction, right? If somebody sneezes at you.
Well, in that case, you have to consider the entire thing, in opposite direction. But the similar physics should
then the efficiency will be different, right?
Efficiency will be, no, because, if you take the force acting in the opposite direction, then, okay, yeah, in that case, efficiency will be different because you face the hydrophobic wall. Yeah.
Yes. Okay.
Yeah.
Okay. Are there any other questions? Okay, if there are no other questions, let's thank Joydev. And thanks, Joydev. And we'll move on to the next speaker, Manas